Hi, my name is Saikat Basu. I'm an associate professor at South Dakota State University. I apologize for not being there in person. Uh, After this recorded talk, if you have any questions, please feel free to send me an email. And my email address is on this screen, and it will also be there on the last slide of this talk. So the topic of my discussion today is how pox viruses spread. And in that context, I'm going to look at two examples, smallpox and mpox. The last person to die from smallpox was this lady named Janet Parker in England. She was a photographer at the Birmingham Medical School. Her office was upstairs from the lab that housed smallpox samples. And after her death in September 1978, the investigation that followed showed that there were some faults in the ductwork that passed through the smallpox lab. The same air ducts also serviced the space where Janet was working, and it was proposed that she got infected through air currents. Now, this theory was objected by at least some parts of the scientific community even back then. Now, in this work, we are going to develop the microscale cross-cutting mechanics of this kind of inhaled transmission and then evaluate whether this could have been possible in this particular scenario. The model that I'm going to describe is fundamentally pathogen agnostic in nature. So in CT-derived anatomical airway geometries, we are simulating breathing, we are simulating particle transport. These are particles that could be thought of as aerosols and microdroplets that carry pathogens into our airway. And then we are connecting the fluid dynamics outcomes with data from other disciplines like virology and immunology to quantify different metrics related to infection transmission. Now, this particular framework in this work is being applied to smallpox. So why smallpox? The first reason is we have a lot of data in literature on smallpox, so that helps us extract the virological inputs that we need for our model. Secondly, whatever translational metrics that our model can quantify, for example, what is the critical exposure duration for initial onset of smallpox, we can compare those predictions with the known data from smallpox literature. Now, if that comparison works, we can then apply this kind of pathogen agnostic framework to other viruses of the same family. For example, the ones that are more relevant today, like mpox. Now, in this work, we have reconstructed two anatomical airway domains that extend till the tracheal base. We have simulated two inhalation rates, 15 and 30 liters per minute, to model steady and relaxed breathing conditions. The airflow is replicated using LES scheme with the subgrid scale dynamics resolved through kinetic energy transport model. Against the inhaled ambient airflow, we are numerically tracking particulates in the form of monodispersed sets with diameters that vary from 1 to 50 microns. We are especially figuring out the deposition rates of these particulates along these red regions in the airway, which are the main initial infective sites for smallpox in the respiratory tree. The bronchial region is also infective, so we are also recording the particulates that are escaping through the tracheal outlet and moving in downwind into the lung cavity. So with this, before we show the actual simulation data, we are also following some gold standard practices in terms of the fidelity of our simulations. So this is the grid sensitivity analysis that we have performed. So the meshes in these domains, they comprise graded unstructured tetrahedral cells in the bulk, along with 4 layers of very fine prism cells along the walls to capture the near wall particulate dynamics. So we have looked at 5 cases with 4, 5, 6, 7, and 8 million tetrahedral cells in the main mesh. And we have tracked this flow outcome resistance in all these meshes. We see that the trend on resistance variation becomes stabilized as the mesh gets finer, and we have eventually picked the one with 6 million as the mesh in which we have carried out the rest of this analysis. I will give you a link to our very recent paper on this, which has more details on this, on the mesh refinement study. But let me move on from here. So these are some sample visuals of the simulation domain. On the very left, we have the airflow velocity streamlines. The second panel shows the vorticity magnitude contours on a midsection that passes through the city reconstruction. It is quite interesting to explore the vortex dynamics in the space just downwind from the laryngeal constriction. And that is where the vorticities peak in terms of their absolute values throughout the airway. The third panel shows the pressure field and on the same midline cross-section. And on the fourth panel, I've shown some representative, very fine particulate trajectories. In fact, all of them over here escape through the tracheal outlet and are moving into the lower respiratory tract downwind. Now, to ensure the reliability of our simulated findings, we also decided to run a very basic level experimental validation. So we have 3D printed this entire airway for the first geometry. We have developed a glottic plug that we can separate from this laryngotracheal domain, and we can measure the particulates that have landed at the glottis and compare that with the corresponding CFD data. The experimental technique that we had used for this was with UV spectroscopy, where the solution that we sprayed into this domain, or rather we nebulized into this domain, they carried some caffeine markers. We also replicated the inhaled airflow by using a pressure pump. We gathered the particulates that ended up at the glottis. We collected them and we measured what was the deposition rate in the experiments, and we compared that number with the CFD ones. Overall, we got an agreement that ranged within 80 to 90%. So the deposition in the experiment was around 80 to 90% of the CFD predicted deposition at glottis. This slight difference makes sense because as we took the glottic plug out, we did lose some very fine particulates that had settled along the edges of this cavity opening in the plug. So with this zeroth order validation, let me now move into the actual simulated data. So these heat maps show the deposition rates at the infective tissue sites, the tissue sites that are infective towards smallpox, for the different flow geometry combinations. So we have 2 geometries and 2 flow rates. So overall, there are 4 cases. In all of them, we see a certain homogeneity. We see that for all of them, if the particulate size is less than equal to 9 microns, the deposition efficiency at those infective regions are at least becomes at least 30%. So based on this equivalence across the flow geometry combinations, we have averaged the deposition rates into this quantity called eta. So this is the percentage mean deposition at the infective sites for each particulate diameter d. Remember that these are monodispersed particulates, so we are getting the data for each d value. And this eta is being averaged from this eta k, which are the corresponding deposition rates from each of the flow geometry combinations. Now, once we have the basic flow outcome data from the simulations, we now need to know what is the size distribution of particulates that one is going to inhale. So for that, we have used respiratory ejector data that have been reported in literature in terms of their particle size distributions. So from this paper, if we process the data, we find this plot which gives the count of particulates of each size that after being expelled got dehydrated because of environmental dehydration and are now being inhaled by the exposed subject. So this gives the counts of particulates of each size inhaled in each minute of exposure. So that being n in, we can then quantify this parameter called sigma, which is the net liquid volume depositing at the infective tissue sites in each minute of exposure. So once we get this parameter from the flow physics, the next step is to connect this flow outcome with the cross-disciplinary parameters. So the first thing that we need to know is the ID, which is the infectious disease of the pathogen. This quantifies the minimum number of variants that can instigate an infection, and it is measured in the form of PFU, or plaque forming unit, which is the number of plaques formed by a viral sample in a host monolayer of cells. So a higher PFU would imply that more virion units are needed for the infection to get launched in a particular host, in a particular exposed subject. The next cross-disciplinary parameter that we need is this VL, which quantifies the concentration of variants in the liquid particulates that are being inhaled. The next parameter is P, which is the potency of these variants, as in the success rate of these variants to start an infection in the exposed subject. So once we have these three specific parameters, ID, infectious dose, VL, viral concentration, P, potency of the variants, we can then quantify this term tau C. Tau C is the critical exposure duration over which an infection might get launched in an exposed subject. And this formalism gives tau C in hours. Now, once we have this, to get the real numbers, we need to get specific data on ID, VL, and P. For us, it was relatively straightforward to get what is ID, or the infectious dose, and the P, which is the variant potency. What stumped us was the VL, because we needed data for fresh samples. And smallpox has been declared eradicated back in 1980. So after a lot of search, we eventually came across this series of papers from the 1970s. So this group, they gathered data from a big number of smallpox subjects. They collected throat swabs, and that data gave us that fresh sample numbers. So we processed their tabulated data, and this is our plot. And we figured out the mean viral concentration from these samples. So these are 147 samples coming from 32 subjects over a time window of 14 days. And these are all hospitalized smallpox patients. So overall, this VL, the mean value is 10 to the power of 2.952 PFU per ml. So now we have all the specific numbers for the cross-disciplinary parameters. We can then plug this back into the mathematical form for tau C, which is the critical exposure duration. So this is what we do next. So these are the plotted tau C trend lines. So the upper trend lines are for lower values of P, which implies that the variants are less successful at launching infection, and hence the critical exposure duration could be longer. Especially focus on this red trend line, which is for a p-value of 63%. This trend line gives tau C estimates that vary between around 1.8 hours to around 17 hours. And these numbers match almost exactly with previously known estimates for smallpox that comes from this probabilistic Wells-Riley model for spatial transmission. They had used empirical estimates for inhalation, and for a probability of infection 63%, which is what it is for one quanta of pathogens, they found out that the range of critical exposure duration should vary between similar 1.8 to around 17.7 hours. So with this cross-technique validation, we would go a step further. But before that, the overall critical exposure duration in our work right now is typically between 1.1 to around 18.6 hours. So now moving a step forward from here, we are now considering viral evolution as well by perturbing the viral concentration, specifically the index here, So the beta mean is being perturbed by plus minus 0.5. As a result, the overall critical exposure duration now ranges from 0.35 hours, which is approximately 21 minutes, to around 60 hours. We have also extended the framework to mpox, which is more relevant today. And again, for mpox, we needed the specific cross-disciplinary values. So the VL mean for mpox is 10 to the power of 2.92 PFU per ml. For that, the critical exposure duration ranges between 24 and 40 hours. We have again considered viral evolution by perturbing the index on this VL formulation, and as a result, the overall critical exposure duration ranges between around 7.6 hours to around 127 hours here. So overall, in conclusion, we see that this model that we have developed by integrating fluid dynamics in complex physiological domains with virology, with immunology, does give us realistic estimates for critical exposure duration that have some level of cross-technique validation. The typical exposure durations for smallpox from our work is between 1 to 19 hours, but it could be as short as 20 minutes based on viral evolution. Now, this sounds very plausible if you think about the patient zeros example that I related at the very beginning. But more importantly, now moving into mpox, there the critical exposure duration is typically between 24 and 40 hours. And sometimes, based on the evolution of the virus, it could be as long as 130 hours. So overall, this 24 to 40 hours is expectedly longer than the typical exposure duration that we saw for smallpox. So the chances of inhaled transmission is less likely for mpox, true, but still these physics-guided numbers should help us if we have to come up with some kind of mitigation protocol in case of an outbreak. Now, this work is currently going through second round of peer review, and hopefully it would get published very soon. But right now, it is also available on medRxiv. This is the QR code for my lab's website. Here is my email address. If you have any question, please feel free to shoot me an email. Here are my PhD students, Mohammad Yasin and Mohammad Akash. He recently completed his PhD and is now a postdoc at Florida State University. So they were in charge of all the simulations and modeling to make sure that we use the cross-disciplinary parameters reliably and robustly enough. We are collaborating with Dr. Chakraborty from Fractal Therapeutics and Dr. Julia Rebecca Port, who is a group leader at the Helmholtz Center for Infection Research in Germany. So with this, I would end. I would look forward to some emails and some questions from the audience. And I again apologize that I am not there. Thank you so very much.